Well, good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Mastering Oncology Immunotherapy Clinical Trials, the Smaller Biopharmaceutical Perspective. My name is Andrew Jernan, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you are involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen, and if you require any assistance, please contact me at any time by sending a message using that chat panel. And at this time, all participants are in a listen-only mode. And please note that this event will be recorded and made available to you for future download. And now, I would like to introduce our speakers for today's webinar. Dr. Andrew Zupnik is the head of strategy for the Oncology division at Novella Clinical. Focused exclusively in oncology research for more than 15 years, Dr. Zupnik provides strategic guidance to the oncology business development and operational teams, as well as a consultative and account oversight for oncology customers. Additional roles at Novella include development of custom protocol-centric solutions for new oncology projects, training to internal staff on drug mechanisms and industry trends, and the pursuit of new initiatives for the Oncology division, including its recent subspecialty focus in immuno-oncology. And your second speaker for today, Dr. Julia Lawrence, is a board-certified medical oncologist with 18 years of experience in clinical oncology and cancer research. She was a program director for the National Cancer Institute for 3 years, where she worked on biomarkers for drug development. She worked in the private sector for clinical practice and then 13 years in academia at Louisiana State University and Wake Forest University. Dr. Lawrence has conducted clinical trials in the treatment of breast cancer and cooperated on clinical trials for colorectal, lung cancer, and prostate cancer. She has also developed clinical trials to monitor and prevent the late effects of cancer treatment to the cardiovascular, central nervous system, and hematologic system. And now it's with my pleasure to hand the microphone over to your first speaker for today, and that's Andrew Zupnik. Andrew, you can begin your presentation when ready.
Thank you, Andrew, and thank you everyone for joining us today. Good morning, good afternoon, and from where I see some attendees joining us, good evening. It's a pleasure to be with you and appreciate you taking an hour out of your very busy day. I know everyone has a lot on their plate, and we hope to keep you interested and engaged throughout this hour. As Andrew said, please submit questions as we go, and we will try to customize the talk and address some of those questions at the end. So with that, wanted to dig straight in on the next slide. That wanted to set the stage a bit around the intent of today and why we are here, hopefully to share some insights with you around the what we're calling the exciting area of immuno-oncology. So Novella Clinical is an oncology-focused CRO, and we have another division that focuses in medical device and diagnostics of over 1,000 people globally. And as part of our business model, we work almost exclusively with small to mid-sized biotech companies. Myself being a researcher, as Andrew said, and a bench scientist by training, I get the distinct pleasure of seeing almost every oncology protocol come across my desk. And we have over 150 active oncology trials today, and have launched almost half of those in the past couple of years. So quite a nice portfolio that we get to touch, and certainly appreciate the ability to work with our biotech client base and partnering with them on what we will talk about today as some pretty exciting and novel technologies. And you can see on the slide there, it's been fun to be part of that space because the model has certainly been in the industry that biotech is driving a majority of innovation. And you see this almost every day that's become pretty standard from a large pharmaceutical perspective around mergers and acquisitions to sort of build and grow their pipeline whereas a lot of the novel technologies are coming out of really the startup space. And that's, for us, really the fun part of getting to work with very small to virtual, to well-established biotech companies, and the very first in human applications of some novel science. And you can see we're calling this the immuno-oncology craze, and that's why we're here today to talk about what's been going on in the industry, what we're seeing now, as well as what's coming up, and then from a smaller biotech perspective, what that might mean as far as challenges and considerations to think about as you're developing and moving into this space or developing along your pipeline. There are almost 100 or 500 active immuno-oncology studies today from some database searches. Almost a third of those are specifically combinations with checkpoint inhibitors, and I bet if you went back a couple of years, you wouldn't see that much anywhere near that level of activity. So it really has exploded onto the space and become quite an interesting evolution for the industry. And then capitalize on the right, we just wanted to highlight that we're excited to be working actively in a lot of these technologies and therapies. So you can see we're working in CAR T today, we're working with dendritic cell vaccines, certainly quite a large number of combination checkpoint inhibitor trials. I think almost half of the protocols that have come across our desk this year have been looking at some form of immunotherapy in novel immunotherapy or in combination. And as well, it's been exciting for me. My research interest back in the lab was around P53, the tumor suppressor, and still waiting to this day, 40 years or so, to see a drug targeting that space make it onto the market. But it's been reinvigorating some what has been tested in the past around gene therapy, oncolytic viruses. I think the influx of capital has certainly been nice to see a benefit that people can finally move along what we've known from a scientific perspective for quite a while. And then, of course, there's really what have been well established, and we'll talk about some of the early antibodies, which are immunotherapy approaches, and what we're now seeing as next generation or next generation technologies. We're getting to touch some bivalent and tetravalent antibodies, as well as next versions of antibody drug conjugates, which have been on the market and well established over the past few years. So with that, I think we wanted to get the audience engaged a little bit and move it into our first polling question. And Andrew, I'll hand it back over to you for that.
Great, thanks for that. We certainly do have the first polling question of the day up on your screens, and you can vote on this in real time by clicking on your screen, and I encourage you to do so. The first question we have for you: what is the most important benefit we've seen with immunotherapy products? Is that metastatic melanoma no longer universally fatal? Another option there, potential for improved safety profile. Your third option, a potential long-term survival with retraining of the immune system, or all of the above. That question, what is the most important benefit we've seen with immunotherapy products? Looks like most of you have voted on this question. So I'll close polling now and share the results with everyone. And overwhelmingly, not surprisingly, 71% of you said all of the above. 19% said potential for long-term survival with retraining of the immune system. And with that, I'll hand the mic back to you.
Thank you, Andrew. And moving along, that was purposely designed to be a softball question for you guys. I think the intent was all of that is what's contributing to really the excitement and the potential benefit of immuno-oncology. I liken this back to the time of perhaps around angiogenesis, where there was a lot of promise around an impact in a wide range of tumor types and a wide range of patients. And I think that's what really comes down to from our perspective. I can certainly tell you for everybody working at Novella and all of my colleagues that I have in the industry, we feel very passionate about the oncology space. We're all here, in a sense, for a reason. As Andrew said, that's all I've ever done in my professional career, and that's all I'd really like to keep doing, because I don't know anyone who hasn't been touched personally by cancer. It's it's a daily sad story that we hear all the time, and why we're all here trying to make a difference and impact even to the smallest degree. So I think that speaks to the potential benefit and why it's become such an exciting area and receives so much attention. It's not just in the news and in our industry media, but I mean we're seeing immunotherapy becoming mainstream news. I'm getting newspaper clippings emailed to me from my family members all the time. Have you heard of this? Have you seen this? It's really getting out there and driving a lot of interest from the general population. We're seeing a benefit with the early drugs that have been approved, and that's really been what's driving the space. You can see on the slide there the initial data from ipilimumab, which was sort of what we're thinking of as the first checkpoint inhibitor approved from BMS. That in advanced melanoma there was almost a doubling of survival, and that was one of the polling questions. Is that now with some of the newer technologies, there are treatment options out there which has made this disease much more tolerable in a sense. We're nowhere near a cure. We still have a long way to go, but it's no longer as significant of a downward spiral, so to say, for some patients. And then some of the newer checkpoint inhibitors, pembrolizumab, nivolumab, have burst onto the scene and are getting used all over the place. And I think one of the nice things that we're seeing is that some of the overall survival data is still not even out there. And so we're getting approvals based on surrogate endpoints like progression-free survival. And you can see on the slide, pembrolizumab had a doubling compared to chemotherapy, and that's a significant result. And that's hopefully translating through to real survival benefits to patients, that we are still waiting for those readouts on the trials, which means that the drugs are working and patients are living much longer than we've seen in the past. And we have very rapid new approvals in lung cancer and renal cell cancer. And then, it seems like every industry meeting, there's exciting new data published or announced as recent as AACR. A few weeks ago, I think nivolumab results in the Checkmate 141 trial were talked about in advanced head and neck cancers. And again, we're seeing some significant impacts to patients, which are driving this excitement. And then when you even look at some of the subpopulations, which we will talk about, how to target some of these therapies and what this means from a genetic perspective, when you're looking at viral status or even biomarker status in the head and neck patients, it was even more of a pronounced effect. So all of that translates to making a difference in the cancer space, and of course the financial component follows along. You can see on the slide there, some estimates are saying the immuno-oncology space is going to be over $40 billion by 2020, and potentially half of the overall oncology market. So all of that connects nicely together, and I think it's been exciting to see the funding component following along and driving a lot of this innovation and excitement. So with that, moving to the next slide, wanted to start talking a bit about different immunotherapy approaches, both what are out there as well as what's coming up. And so I will hand it over to Dr. Lawrence to talk you through those pieces.
Thank you, Andrew. So proceeding onward then, I am going to talk to you first about different classes, as Andrew indicated, to give some indication of where we see this field going. Anti-tumor antibodies are, the original was trastuzumab and rituximab, which have significantly contributed to survival in breast and lymphoma, and are more of the simplistic antibodies within this field. The next was the antibody drug conjugates, and the ones that are on the market are brentuximab and TDM1. We see a lot of potential within this field. Certainly as we discover new tumor-associated antigens, new antibodies will be defined as well. And also the payloads or the cytotoxic agents that have been used thus far are fairly limited. And there's a lot of potential in this field to move to getting more payloads or more cytotoxic chemotherapy to attach to the antibody that really makes sense and derive from really our vast experience in using cytotoxic chemotherapy for decades. And then thirdly, another moiety within the antibody drug conjugate is the linker of these two, the antibody and the drug. And these can be tailored to be cleaved within the cell or extracellular, and there is reason to optimize one versus the other about where you want your cytotoxic chemotherapy brought in proximity to a tumor cell. And the last class is the one that's represented in the cartoon, which is the bispecific antibodies. And these are two-pronged antibody, and one prong is to the tumor-associated antigen, and the other is to a T cell receptor. So in effect, bringing T cells into the microenvironment, which clearly then would be potentially complementary to some of the other therapies that we're seeing in the other classes. So, and then vaccines have really made the most significant contribution to cancer prevention, and that using vaccines for the infectious etiologies of cancer and having long-term downstream effect of decreasing cancer incidence. And that is the vaccines for HPV and HBV. The challenge is still, and the work still to be done with vaccines, is that these diseases are more typically in resource-deprived areas, and so applying more global immunization to really decrease the incidence of cancer is still work that has to be done on a global level. Therapeutic vaccines are vaccines that are then targeting cancer that has already occurred, and these fall into different types. Peptide-based cancer vaccines are those that are against like glycoproteins or other proteins that might be shared across different cancers, like MAGE, and MUC1 is another example. So be developing a vaccine that could potentially be treatment for various different cancers. And these haven't gained a lot of traction just yet, but are still being looked at for development. Cell-based vaccines are, in fact, harvesting cells, white blood cells from an individual, and altering them and reinfusing them, or autologous T cells being reinfused. And Provenge has been approved for prostate cancer for this indication, in reinfusing modified dendritic cells into the individual. And then virus-based vaccines are utilizing a virus that is not terribly harmful, such as the herpes simplex virus, and altering it so that it creates activity at the cell, at the cancer cell. This particular one, TVEC, the herpes simplex vaccine, has been altered so that it replicates within a cancer cell and not within the normal cell, and also has a vector that would secrete GM-CSF, and in thus doing bring dendritic cells to the site of activity for an added anti-cancer effect. Adoptive T cell therapy is potentially the most complex of these approaches, in that you're essentially taking cells out of the host, the patient, and reinfusing them. And what's occurring is that you're overcoming tolerance by using the natural ability of the immune system. And approaches to this have been tumor-infiltrated T cells, which have been around for some time, as well as the dendritic therapy vaccines, which I just mentioned on the previous slide. But the most excitement exists around CAR therapy, and CAR therapy is chimeric antigen receptors being modified within T cells and then reinfused. CAR T cell therapy is been shown promising results thus far in CLL and in ALL. And what is particularly remarkable, for example, in ALL, is that with CAR T cell therapy directed at CD19, not only there was a very high complete response rates for patients that were previously treated, but there was the persistence of the CAR T cell therapy, CAR T cells within the patient's blood for up to 1 year post-treatment, suggesting a long-term effect and a long-term tumor control. Other cells that are being manipulated externally are the CAR NK cells, which might be looked at as complementary with these two cells of activity. As a medical oncologist who has been using chemotherapy for my career, to hear about the complexity of adoptive T cell therapy is really something to carefully consider when we're thinking about our clinical trials. I'm just going to lead you through what occurs to the patient and the system, while and how it's going to be quite a challenge to get this applicable to multiple sites. So, and white blood cells need to be apheresed from the patient through a large bore catheter. So quite commonly that's a central line. Patients who have leukemia might more typically have a central line than a patient with a solid tumor, but quite often these central lines would have to be inserted just for the leukapheresis procedure. The cells are then harvested and taken to a central site, and then they're activated, for example, in the ALL therapy that I mentioned that was developed at Memorial Sloan Kettering, the cells are activated with antibodies to CD3 T cell antigens, and then they're also transduced with a CD19 gene as well, a CD19 complex with a T cell receptor zeta chain, and that helps to propagate the genes and propagate the cells and get them poised to attack the CD19 once reinfused. So the cells are expanded so that a sufficient pool can be reinfused, and then those modified cells are brought back to the patient and reinfused into the patient, sometimes with chemotherapy. So at this point, a lot of these studies have been done at single sites. Just now we're seeing them being expanded to multiple sites. And of course, just that leukapheresis and sending out a specimen could add complexity, and it's important in designing the clinical trials that you develop a system that can be exported and used long-term when the successful product is found. So checkpoint inhibitors, as Andrew mentioned, have really garnered the most interest and the most excitement, and with good reason. They are targeted antibodies to antigens present on a T cell that's inhibiting an activation is inhibiting immune response to the tumor. So the ones that exist today are anti-CTLA-4, which is ipilimumab, and again showed incredible responses with multiple myeloma, and then the next generation was even more improved, that to anti-PD-1, which is nivolumab and pembrolizumab. Interestingly, although the overall response rate improved and progression-free survival has improved when these two agents or classes are added, we haven't yet seen overall survival and some of the overall survival improvement with the combinations. And some of the early results from AACR suggested that potentially they might not necessarily be improving on overall survival, although they're improving a response rate, kind of suggesting that we might need to look at something more than just response rate to decide on these combinations. Other excitement around this class is what other combinations should be used. Traditional chemotherapies are a really important thing to look at because they change the tumor characteristics and might, in essence, make it more of a setup to have an immune attack. Even low-dose chemotherapy, Cytoxan, or radiation might change the integrity of a tumor so it's more vulnerable to a checkpoint inhibitor. And one thing that's been recognized is the antigen presence on a tumor cells, or neoantigens, are directly related to the DNA mutation rate within a cancer cell. And so that the more mutations, potentially the more neoantigens that exist. So the molecularly targeted field or therapy field will clearly continue along with the immuno-oncology field because these two fields are becoming tightly linked, and really sensible combinations are to emerge as we understand more of the correlation of the molecular targets that are going along with these immune response patterns. And then as Andrew mentioned, the really big promise is that now we're seeing flattening of curves for diseases that we did not see flattening of survival curves. So there clearly is a subset of patients who are receiving these checkpoint inhibitors who are not progressing over the years that they've been followed, and that is highly suggestive that potentially either a cure or some real significant disease stabilization has occurred. This cartoon represents what you expect to see for this field, in that the only checkpoint inhibitors that have actually come to the clinic just far are the CTLA-4 and PD-1, but those in development are along the cartoon in this, as well as activating receptors will clearly be contributory, and sensible combinations of these will certainly emerge. And I'm going to turn it back to Andrew.
Thank you, Julia. And I think this is the part where we'd like to shift the discussion. Thanks for taking us through what's out there now and certainly what's coming up. And there are a wide range of even classes within immuno-oncology. So I think that's what we wanted to talk a bit more about, what the considerations may be for our small to mid-size partners that we see and that we're working with. And so moving to the next slide is a bit of a summary of some of the challenges that hopefully with the audience here being specialists in that area, I'm preaching to the choir probably to all of you. But from my perspective, the top bullet there is yes, we have innovative drug designs. This is all very novel science. Some of it is very complicated. And I think that can be a little bit scary to some folks. Myself being an oncology bench scientist and the cell and molecular biologist, definitely is wonderful to see us harvesting what has been known from a biology perspective for quite a while. But the immunology space is a bit foreign. And so I think the challenge that this whole boom creates is, for folks who've been working in oncology, I can at least tell you for a long time, the immunology and oncology specialties have not traditionally worked together. I've had to go back and get some, undergraduate textbooks on immunology and to look up some of the details and just get more familiar. So I think it's a challenge that it creates, that there's a novel space. There's a learning curve that certainly people who are familiar with an oncology background and oncology development and protocols and assessments as well, that we will talk through some of those key pieces which are important to kind of retrain and build that learning curve. And of course, from a smaller company perspective, it's, we understand the limitations. We don't have all the firepower of a large pharmaceutical, all the resources, all the infrastructure, and certainly the financial resources. So there's a need to lean on partners and external input, not just CROs, but of course, building scientific advisory boards, pulling in some key opinion leaders, and even folks who are up and coming in this space. We found a lot of interest in new medical oncologists and immunologists looking to get into the area and create a name. There's a benefit in outsourcing and using that group to lean on your plans. The third bullet is there, the financial management. I see this every day, and from a small perspective, obviously this is potentially the most critical component, that you may have one product in the market and there are it's your baby. All of your efforts are hinging on its success and a desire to move as quickly as possible. It's a different perspective when you have VC firms breathing down your neck saying, where's our data, where's that first patient, where's that site milestone, and needing to move quickly. The stakes are high. And I think that's why it's important to really understand all of the considerations in planning so that you don't run into potential risks and impacts to timelines, because ultimately time equals money. And with the rapidly shifting landscape, it also equals a window for the competition to catch up. And that's sort of the next bullet there is the standard of care. It's shifting very rapidly. I'd say historically it was a little easier to understand in a given indication what a current standard of care is and what might be coming up in the next couple of years. It's well known. There's a large pivotal phase 3 that's going on with this company. We think that might get approved by a certain time, and to plan accordingly. It's becoming different with immunotherapies these days because there's so much going on. And of course, with the regulators, I think getting pretty well on board with the desire to move products through the clinic and out onto the market and into patients' hands, ultimately, that have a significant benefit. It's been great to see, but it's a challenge that it's harder to sort of forecast the future. And so it is becoming important to understand what the potential market access may be. From a smaller biotech perspective, that was rarely something to have to worry about in a first-in-human trial. And now it is something to think about, in this protocol design and even my drug, how does that fit in with where things may be 1, 2, 5 years down the road? Is it even going to be relevant at that point with so many people going after this space? That's a tough question, and certainly something to think about as part of the pipeline and protocol developments. And that connects with the competing trial landscape. With so many trials, as I said at the start, going on out there today, of course, there are only so many centers in the U.S. and of course, ex-U.S. That are comfortable and familiar and, for clinical trials, have the patient volume to support the timelines that are needed. So, we've certainly seen an interesting challenge on the feasibility and accrual modeling around timelines. I can't point to specifics, but I've seen some sites, some groups that we work with very regularly in phase 1 oncology side of things showing enrollment logs and showing us the data that because this is out there in the public domain and becoming mainstream media attention, patients are seeking out these trials. Patients are refusing standard of care. They're refusing chemotherapy. They're saying, nope, I don't want that. I know what the results are going to be. I want to try something new. I want an immunotherapy. What trials do you have that I can work on? And that's a very different space. The patient side of things and their caregivers are very educated. There's a lot of information out there, although it's hard to weed through. I think that's changing what we've seen around how to plan for certain trials. And it's a good problem to have in some cases that it's still yet to be seen on any trial, but we have seen some phase 1s even with dose escalation with so many sites and investigators looking to be involved that they can move very quickly. And that's different and a great problem to have, and it helps move along the development program faster, but how do you model and plan for that accordingly is the question. And then, of course, the last bullet on the right there is the combination therapies. We're seeing, I think, probably half of the oncology protocols we've seen this year at Novella have had some form of combination in mind, or as the next steps in development. And so, from a small biotech perspective, how do you get access to that? Say we're talking about a checkpoint inhibitor. Do you look to partner with BMS or Merck? And I think Merck has been certainly more out there as far as getting pembrolizumab into other people's hands and allowing them to test different combinations and indications. I think BMS is a little bit more conservative on the partnerships that they're entering into. So if you don't have that luxury as a small biotech company, how do you get access to this drug when they cost $125,000 plus for a treatment regimen? Do you plan to reimburse for it? And especially when you're considering from a global perspective, these drugs aren't necessarily approved everywhere or even used everywhere. And how do you factor that into your planning? So the bottom line is it's important to assess the expertise specific to immuno-oncology. It's different from just oncology is what I'd say. And to really understand what that expertise can bring to the table to help you in those planning stages. And, we love an opportunity to get involved that early to help share our thoughts and insight and hopefully bring some benefit to those plans. So that, hopefully that's where we wanted to move to the next section, which is sort of the 5 challenges to consider in developing an immuno-oncology clinical trial. And for those of you who are multitasking out there, I do this myself. Hopefully this is the real meat of our discussion. Come back and join us. We wanted to hand it over to a new polling question to get your thoughts on what is of most interest to you to help sort of shade the rest of the talk.
That's right. Thank you very much. We do have the second and final polling question appearing on your screens right now. And again, you can vote on this by clicking on any answer there that you see fit. So what do you consider the most significant challenge when developing immunotherapy products? Your options there are protocol design, recruiting the right patients, dosing, managing adverse events, or finding a pronounceable name with less than 5 syllables. So again, you can vote on this in real time. I encourage you to do so. What do you consider the most significant challenge when developing immunotherapy products? Looks like most of you have voted on this question, so I'll close polling here momentarily and share the results with everyone. There we have it. 48% of you said protocol design, 29% said recruiting the right patients, 10% said dosing, 9% said managing adverse events. Just 3% of you finding a pronounceable name with less than 5 syllables. With that, I'll hand the mic back to you.
Thank you for your 3%. I appreciate the comedy there. And thank you. That really helps shade the topics we wanted to walk through with the rest of the presentation. And seeing the most interest in protocol design, it's fantastic that we've brought that up first. And I think there are quite a number of things to think about here, and there's no one-size-fits-all solution. But I wanted to touch on a couple topics around the protocol components because, as I said, there are new pieces to consider. And the first is certainly around the interest in looking at earlier looks at efficacy, we have seen a desire by the regulators to move products forward. The FDA has been very true to their word with breakthrough designations, accelerated approvals. Some of these immunotherapies have been getting approved with surrogate endpoints almost out of a phase 1 design. And that's becoming more common and something to think about when planning a first-in-human trial. I would say the classic 3+3, look for an MTD and stop design is gone. Some people are still pursuing it, and I see that there's benefit in the stepwise approach, and especially when there's limitations of funds or resources. Those are important to consider. But I will say it's becoming much more common and accepted to at least look at investing in expansion cohorts. I mean, we know, if you know the science of your drugs, you know which indications you ultimately want to go after. You know which potentially biomarker-driven patients to go into. And I think it's becoming quite appreciated, in fact, that let's just go straight into that population right out of the gate. Yes, you want to make sure that we're safe. That's the intent of a phase 1 trial, to find your optimal dose. A lot of these immunotherapies, you don't have the toxicity profile of classic chemotherapies or some other drug classes. And so it becomes more a question around finding the active biological dose or the recommended phase 2 dose. And those are different questions when designing a protocol. So I'd say it's becoming quite standard to at least have expansion cohorts and what some people may call a seamless phase 1-2 design, or phase 1-2A. Everyone has different definitions. And then to even go into a couple different indications all in one protocol. Some people are getting very aggressive, and I think that it's still yet to be seen how the regulators may respond to a seamless phase 1-2-3 trial. But the largest phase 1 we've launched this year has been 250 patients. That is a huge difference from the complexities of running a 21 patient 3+3 trial. And so it's becoming more common, and there are ways to be much more creative in how you're building your protocols to go straight into looking at that early look at efficacy. I remember when looking at FDA approvals for 2014, this is still 1 year out of date, but any new molecular entity, I think only one of them that was approved with overall survival as the endpoint. The FDA has certainly gotten behind the benefit and value of surrogate endpoints like progression-free survival, and these immunotherapies are even pushing the envelope even further around data that is very early, yet so significant that the government is saying, yes, we support this and we want to get this out there in patients' hands. And I think that's fantastic, that we're really looking at what I see as basically removing the old phase 2 component of a phase 1-2-3 development plan. It's becoming common to go from a seamless phase 1-2 and then to a registrational trial, and that's what you have to lean on with some accelerated approval, and then you look at the overall survival endpoints on an ongoing basis. But it's enabling these exciting therapeutics to get out there and help patients. And so that's a different from looking at your design. And the second little sub-bullet there is because there is a potential to look at efficacy right out of the gate. And I think I heard sort of third hand that there has not been an oncology product approved that did not have some hint of efficacy in phase 1. So it's becoming standard especially in the solid tumor space, when you're looking at usually using an imaging component as your primary efficacy measure, that people are looking to collect and hold that and use that information, that data, potentially hold those reads for a later stage, and when you might want to go back and look at those in a central way. So the next sort of box there is that with these protocol designs in immuno-oncology, there's very different effects on a tumor, and Dr. Lawrence will talk a bit more about that from a patient and a clinical perspective. But you have different endpoint measures that have been developed. There's IRRC, IR-RECIST, you have IRANO and INANO for brain cancers, you have modified RECIST for certain liver indications, and those are different. You have to understand the impacts of how those calculations are done and build those into your protocol. However, they have not necessarily been vetted and approved from a regulatory perspective yet. I think we'll get there, but today you still have to build in the classic RECIST for a solid tumor endpoint, potentially as your secondary or even primary on phase 1 trials sometimes, in addition to some of the immune-related responses. And that's a challenge to build that in and design your protocol effectively around those endpoints. And then we've talked a bit about the third and fourth pieces there, is that the combination therapies are certainly the way things are moving. It makes perfect sense from a biology perspective that we have targeted therapies which are going after specific molecular defects in a cancer which are driving tumor growth, tumor protection from apoptosis, and other tumorigenesis pathways. And you're combining that with sort of this reinvigorating of the immune system to recognize and attack cancer cells. So that's definitely where the industry is moving, and hopefully where we'll get one day to have a whole collection of options for patients. But that is a complexity around protocol design when you are looking to provide combination drugs. And how do you assess that, especially when you're talking global, which is the bottom piece here, and with these larger phase 1 designs even right into the clinic, the international consideration is something to think about, given the complexity and the competition, especially in the U.S. market. There's an area for going overseas right out of the gate, which can enable you to move even faster. And so when thinking about those regulatory considerations and the combination components, you have to understand, because the market's shifting so rapidly, what the current landscape looks like. The U.S. is probably at the forefront of approvals, and you have the EU, which is very close, but sometimes I'd say averaging about 6 months behind with some of these approvals, and then elsewhere things aren't even approved at all yet. So how do you assess appropriately the current landscape? And even if approved, is it being used? And even if being used, is it being reimbursed? So these are all important questions to get experts on the ground from a regulatory perspective to assess how things look today, and especially when you're planning a protocol potentially 6, 12 months from now, how that might be different. All of those are important questions to take into account. There's no one solution, but it is becoming much more complicated to design a protocol in sort of real time with how the industry is shifting. So with that, I wanted to move on to sort of topic number 2, and Dr. Lawrence, I'll pass it over to you from the clinical perspective.
Okay, sure. So another challenge or consideration when thinking about your protocol development is dosing and measuring response. So typically for the first-in-human study, an animal model would guide on that initial dose. However, with animal models probably being a lot more limited as far as insight into what's a tolerable dose in a human, that the initial dose may be pretty far off of where you think your active dose is. So this brings into the question of that rapid dose escalation, consideration of doing one patient cohorts until a grade 2 toxicity is seen, and then go to the 3+3 design so that you're minimizing the patients at the lowest dosage. Also, in thinking about, that we're not going to be treating to an MTD because it's not a compound that we expect to see toxicity, then typically you'll be thinking about pharmacodynamic or pharmacokinetic markers as guidance. And it's really important in that, if that is the intent, that you get your lab specimens back in a timely way and have some system of batching them and getting them back so that our cohorts can proceed nicely and that patients will be enrolled, and that would not lead to delays when moving to these kind of non-traditional dose escalation patterns. And then measuring response. As Andrew said, this is moving earlier into efficacy, and so you will be left with this challenge of including the immune response assessments with the RECIST. And I do think that there'll be other reiterations of these immune response criteria, and the intent of them is that we are seeing this phenomena of pseudoprogression with these compounds, and what that is that a tumor actually might increase radiologically, and that particular patient may then later have a tumor response with a shrinkage. And we don't want to falsely take off a patient while they appear to have some form of progression, which in fact might be an appropriate cytokine immune response that is what we were achieving or looking for. So that premise of treating past a pseudoprogression or confirming progressive disease is a new challenge as far as thinking about how to design progression-free survival endpoints when you have kind of a delayed confirmation of response. And getting adequate training both within your team and at the site is going to become imperative with that. And as always, we want to get the right patients on our studies, and biomarkers are really becoming commonplace in all early clinical trials, but especially to the field of immuno-oncology. There's some really interesting activity. Even just histologic patterns or immune distribution within a tumor might be insightful to who is going to respond to different immunotherapies. There's been some advances in characterizing tumors as lymphocyte predominant, lymphocyte depleted, but also other patterns such as like where immune cells are distributed, if they're on the margin of the tumor or in an infiltrative pattern. And so all of these are important to think about up front, because we really want to know which of our patients are the ones that are responding to these agents, so that the follow-up trials can be carefully planned. So how you obtain tissues is an important consideration, archival versus fresh. Always make sure that you have some built-in, many patients are going on phase 1 clinical trials, have been on other clinical trials, and those archived specimens may be somewhat depleted. So really think about what specimens you're obtaining and of what value they are. Companion diagnostics, at this point, the immunohistochemistry for PD-1 and PD-L1 is proving quite informative for the checkpoint inhibitors in defining the specific subsets that are benefiting from those agents. So it's now at this point they're being done somewhat retrospectively on the current clinical trials, and eventually they'll have to be done prospectively to really define if they're a predictive marker for response. And so it's important to kind of think about those diagnostics and how you'll develop them in concert with your drug, so that both will be available eventually to define the appropriate patient to be treated. I've talked about how complex this whole treatment paradigm is, and so really think about the patient in your clinical trial design, because they're already taking on the complication of the therapy. So try to bring them the convenience of different assessment schedules and time involved in clinic, because they're already taking on more than oncology patients have had to do before, and collect quality of life data that might support that insight. And really it's important for us to be informational to our patients. As Andrew has mentioned, there's a lot of excitement with this in this field, and it's really important that we continue to define patients that are appropriate for clinical trials. Sometimes because of that excitement, patients who would not otherwise be appropriate for a clinical trial are now becoming very excited about this field. But we really need to keep as advocates of really making sure that patients who are healthy enough to withstand the side effects of immuno-oncology are the ones that are included on the trial. And so reactions of immuno-oncology agents are fairly unique, and consider those in your design. There is now defined pretty much the immune-related adverse events, but I'm sure others will be discovered as we identify new agents. The other reaction or long-term effect of cancer immunotherapy is that it has the potential to adapt to a cancer that continues to mutate. And so that kind of continued response is not something that we've previously dealt with in oncology, and how do we really capture that in thinking about our clinical trial, that it might not be just the same endpoints we've always decided on, response rate and progression-free survival. There might be other things that you might want to add into a study to think about how is the immune response being growing with or changing with the persistence of the tumor. Serious adverse reactions are unfortunately a potential dire consequence of immunotherapy. They have the potential to have limited side effects, but they also have the potential to have dire consequences. As these antigens are identified within the lab and brought to a human study, sometimes the other effects or other sites of these antigens are not fully understood. And that could lead to dire consequences if an antigen is shared with some tissue within the lung and some inflammatory response is set off within the lung, which can be devastating to a patient. So these first in human experiments really have to proceed with caution with that regard. That first line, early stage immunotherapy, may have long-term persistent effects. And so what is the consideration then in thinking about eligibility criteria and where patients are as far as receiving prior immunotherapies, and if you need to be concerned about overlap between the different therapies that they're receiving on different clinical trials. And it may be a challenge to maintain a blind in these studies because of the immune effects. Some consideration may be going to do prophylactic therapies to prevent the adverse effects just to maintain blinds. And the last challenge that we're going to talk about is the patient and site education. So the patient should always be at the center of all of our thinking. And as many patients coming to a clinical trial are fairly sophisticated, that they know that once a tumor gets worse, they should be coming off study. And we really have to do upfront education about the pseudoprogression and that we want to retain them on study even when their tumor looks worsened, not for indefinitely, but until we confirm that it truly is worsened. And also to just use what media is available to you, 21st century, with so much more technology to use as educational tools. And we also want, since we're aware of these potential adverse events, really be upfront about treatment of the adverse events, because we're not seeing that like steroid treatment is really impairing these immunologic approaches. So it's not, I think we should be more liberal about treating the adverse events. And then I've also talked about just the complexity, and it's a challenge for the oncology community and all the way down to really the patients. The practitioners are really with direct hands on the patient, and the training needs to go all the way through the nursing staff and technicians that are talking to the patients. And with these special procedures where product is being phoresed and then sent to another site, we need to make sure that temperature control is maintained and maybe additional support staff brought to the site and good education and education just about these immune responses and safety that is concerned both to the patient and the direct caregivers. And then Andrew?
Yeah, and I know we're getting close to the top of the hour, so really wanted to wrap up the discussion here and then open up for some questions. But I wanted to close by a bit of a altruistic look to the future. Myself, as I've said, being a scientist, it's really thrilling to see the potential and where we are today. Some numbers on the slides there are showing that there's a significant number of cancer drugs in development, certainly active oncology trials. And then, as we talked about at the start, the potential for the immuno-oncology space, as we're seeing on the growth curve, to really take on a predominance of this field. There's a quote which I like in the top right there, which speaks to where hopefully we will be not too far. I'd say in our lifetime that ideally, we will get to this combination approach, as Julia said, using low-dose chemotherapy, which we have very strong, knowledge from decades of use and its toxicity profiles, combined with immunotherapy approach and targeted medicine around genetic mutations, and then potentially these engineered killer cells, all of which are designed for a patient-specific cancer. And we now know and have known from a scientific perspective for a while that cancer is a different disease than most other therapeutic areas. It's a collection of diseases. It's hundreds of diseases sometimes. And so how do you approach that? And that's why we haven't seen cures, is because it changes and morphs over time. And using these combination approaches with immunotherapies that we're seeing today, especially to retrain and almost teach your system to look for these cancer cells and to eradicate them when they pop back up, is really an exciting promise and the future of the industry. I think the ultimate goal in the bottom right is to treat cancer as a chronic disease, something that hopefully you go into the doctor's office, you get a lab, a blood sample taken, and we've been working on some projects with these new liquid biopsies, very exciting technology that crosses into our medical device division, and you can get a complete proteomic profile of your cancer, and then here's your cocktail of drugs that'll work for you, ultimately treating it as a chronic disease that we can stay alive and survive with for as long as our bodies continue. So with that, I think we'd love to take some questions.
Fantastic. Well, thank you very much, Dr. Zepnick. Also, thank you to Dr. Lawrence for her portion of the presentation. Really, really insightful. At this point, I would like to invite our audience to continue sending in their questions and comments, and right now using that questions window. And let's get started. I've already received some questions here. Our first question, what is the trending alternative to the classic 3+3 design?
I will take that one if that's all right, and Dr. Lawrence jump in. But that's a good question. I think the classic 3+3 design is still very common, very comfortable. People know how to use it, how to design around its sites, and their staff are comfortable with it. I think from a smaller biotech perspective, it's interesting. The term adaptive trials has been thrown around for quite a while. I'd say it's been a buzzword for, I don't know, almost 10 years. From the folks that we work with, I'd say at least 90% of our protocols still utilize some form of the 3+3 design. And I'll say it's becoming more common, and that the one model methodology, I think, is called the accelerated titration, which is really essentially single patient cohorts, which you escalate until you see typically a grade 2 toxicity or higher, and then you expand out into the traditional 3+3 approach. That's becoming pretty common, and I think that saves a couple cohorts and a couple patients, and certainly a couple months potentially on timelines, and sites are familiar with that. A lot of people are interested in more adaptive methodologies, and we have statistical experts on staff and partner with some consulting groups which are experts in Bayesian design methodology. But for our client base that we work with, people are interested, but I'd say most folks are not going to that level of complexity. It is a bit of an unknown area for some of our partners, and the fact that you have to have statistical involvement throughout, and there are some reports that it can save time but may not be significant versus the cost. So I'd say most folks are sticking with the single patient cohorts, and then 3+3 designs are still very, very common and comfortable.
Okay, well, thank you for that. Moving quickly now to the next question. Does Novella have experience in basket trials with multiple sponsors therapeutics, or is this a direction you're looking?
Yes, the basket studies, and we can debate about what the definition of a basket trial is. But yeah, I think it's becoming common, as I said, for looking at early efficacy and impacts in first-in-human designs. You have multiple indications, that was what most folks call a basket, and multiple combinations or dose regimens which are being built into protocols right out of the gate. Now, using multiple sponsors is a whole extra level of complexity, and that is something that we're happy to help coordinate, but that's a financial and a partnering component, which it's not our molecule. We can't control any of that. We do, however, have experience with what some people call master protocol designs, and I can't go into the details, but that's very exciting approaches. Vice President Biden was just talking about the Cancer Moonshot program a few weeks ago, and that, I think, component of looking at multiple combinations all in one design is a fantastic approach. There's a lot of complexity, but yes, Novella's the basket trial, at least from a multiple indications with potentially the same biomarker or genetic makeup, is very comfortable.
Okay, interesting. Thank you very much. Maybe we'll squeeze in one last question here. Have you seen any situations where total tumor burden being assessed volumetrically could be considered?
Yeah, I'll take that, Andrew.
Sure.
So I have looked at this field, and the technology is really coming there as far as radiology companies being able to assess things volumetrically. I think the limitation of it at this point is not all the tumors and all the organ sites lend themselves to this, and that really it is kind of more dependent on tumors becoming spherical shapes, and that lends itself the most, whereas many of tumor sites, like in the lung or in the bone, the tumor doesn't really lend itself to these volumetric measurements, although I'm sure in due time this will be there, but I don't think it's there just yet.
All right. Well, excellent. Thank you very much for that, Dr. Lawrence. Also, thank you to Dr. Zupnik for all of those answers. We have reached the end of the question and answer portion of this webinar. If we weren't able to attend to your questions today, the team at Novella Clinical will try to follow up with you after the webinar. And if you do have any further questions, you can write into the email that you're seeing on your screen right now. That's info@novellaclinical.com. And I also encourage you to visit novellaclinical.com/oncology for more information. Thank you, everyone, for participating in today's conference. You'll be receiving a follow-up email from X-Talks with access to the recorded archive for this event, and a survey window will be popping up on your screen. Your participation is appreciated, as it'll help us improve our further webinars. Now, I just sent you a link in your chat box. You can say thanks to our speakers via Twitter using that link, so I encourage you to do so. Thank you, Dr. Andrew Zupnik and Dr. Julia Lawrence, for today's presentation. We hope that you found this conference informative today. Have a great day, everyone.